Hello. So my name is Alexander Hauser. I'm an assistant professor from the Department of Drug Design and Pharmacology at the University of Copenhagen in Denmark. And today I would like to talk about how I'm exploiting data-driven approaches to facilitate GPCR pharmacology and drug discovery in the context of pharmacogenomics and personalized medicine. And specifically, I start with a little introduction on biomedical data, how I'm using data to rationalize research questions, and then I'm going to present some of my recent projects linking computational and pharmacological data on some specific examples. So as you know, there's been an explosion of data in recent years, virtually all domains of the biomedical sciences, from the number of published literature articles to generally databases and resources, genomic data, genetic variation, gene expression, protein interactions and protein structures, compound libraries and whole pathways, not just in human, but a range of model organisms. And we have now access to data at many different scales and resolutions. The NIH and the European Bioinformatics Institute estimated that there is now more than 200 petabytes of processed biological data. Similarly, you might have seen these charts, which show how the reduction in sequencing costs has resulted in an explosion of data, with now many hundreds of thousands of individuals having been whole exome or even whole genome sequenced. And as you also see here on the side, that the main driver of the cost is not the sequencing anymore, but rather the sample collection and the downstream analysis, which I expect to continue to become more and more of a bottleneck. So hence we need to transition into putting the tools into people's hands and to analyze and make use of the data they are generating. And given the wealth of information, we also need to think in how we can best apply informatics and data science approaches on the integration of these diverse data sets to gain new knowledge, with the emphasis on the understanding of human health and biology. And doing so, we need new approaches and the targeted combination in the selected biological system to advance in the pharmaceutical sciences and in drug discovery. So here, I would like to present a project on GPCR pharmacogenomics, and specifically how we can elucidate the spectrum and impact of how a GPCR variant influences ligand binding, signaling, and ultimately therapeutic effect of physiology. And this is important because despite rigorous clinical testing of medical products, do we still reside in times of imprecision medicine, exemplified by how few people benefit from our most prescribed drugs. So that in the future, we hope to tailor selection and dosage of drugs to the individual's composition. What you see here is adverse reactions actually do make up somewhere between the 4th and 6th leading cause of mortality, and 90 to 99% of the population has at least one high-risk variant for a recognized pharmacogenomic gene. And today there is variations in 20 genes that affect now, I think, more than 100 medications that are actionable in the clinic. So what you see here, what contributes to the imprecision medicine is to the largest extent the individual characteristics of an individual, such as age and gender, but also the pathological conditions, comorbidities, polypharmacy, environmental exposure, and lifestyle factors such as smoking and sleep. And of course, and that's what I'm going to focus on here today, is that the genetic contribution to imprecision medicine has been estimated to range somewhere between 20 to 30%, where we can distinguish the genetic factors into variation in ADME genes and to variation in drug targets. And in recent years, there's been quite an increase in both pharmacogenetic and pharmacogenomics, given new tools such as targeted gene sequencing and the advent of the Human Genome Project, but also the advent of larger consortia such as the PharmGKB 1000 Genomes Project and many others. Whereas if you look at the trajectories of the GPCR, there's also been some milestones and important breakthroughs in the complex studying of GPCR signaling, but also given the breakthroughs in cryo-electron microscopy, giving us insights into the receptor G-protein interaction and the different folds of the GPCR. So among the genetic factors that can impact drug response, we can distinguish between variation altering the pharmacokinetics, such as the absorption, distribution, metabolism, and excretion of drugs, and on the other side, the pharmacodynamics, the effects of the drugs mediated by the drug targets, such as nuclear receptors, ion channels, kinases, and GPCRs. And here I'm focusing on GPCRs specifically. And this is because GPCRs regulate virtually every aspect of human physiology, and they're organizing complex activities and communication between different tissues, given a broad range of hormones. And as you see here, from mood, behavior to smell, vision, and taste, GPCRs are involved in nearly every physiological component. So it's not a surprise that GPCRs make up more than a third of all molecular targets of approved drugs. So given that GPCRs are attractive drug targets, we investigated the following questions in our study, which I start here with. So first, how variable are these GPCR drug targets in the human population? Second, are individuals with variant receptors likely to respond differently to drugs? And third, what is the estimated economic burden associated with variation in GPCR drug targets? And the answers to these questions have important implications for personalized medicine that considers an individual's genetic makeup in designing treatment strategies. So as you know, we are all different, and so are our genomes as a result of very subtle differences in our DNA. And it is estimated that on average, between two individuals is roughly 0.6% difference in base pairs from our whole genome. However, there is only about 2% of the genome which actually encodes for the roughly 20,000 genes and proteins, which we refer to as the exome. And the mutations in the exome are expected to have most profound impact on physiology, and this is what we have been focusing here. So we can actually distinguish different types of mutations, such as synonymous, where there's no change in the amino acid composition, missense mutations, which lead to a residue change in protein structure, and loss of function mutations, such as frameshift or indel mutations, which have a much larger impact on protein structure. So here we focus on missense mutations that have lead to a change in amino acid composition, and also we glimpsed a little bit into loss of function mutations. So in order to assess the prevalence, the functional and economic impact due to GPCR drug target variation and variable drug response, we employed a wide range of data types and data resources, starting from all drugs, all approved drugs targeting GPCRs, then the variant data from, at the time, 60,000 individuals, but also de novo mutations from father-mother-child trios, as well as genotypes from the 1000 Genomes Project. On the other hand, functional effect data on ligand binding, G-protein arrestin interface, the post-translational modifications, known variant disease associations, but also clinical annotations of drug variant pairs. And then we have also included sales data from the National Health System in the UK, where we had information about prescription and sales data of drugs targeting GPCRs. So starting with the first question, what is the prevalence of GPCR drug targets to have a missense variation within an individual? We employed drug target data as well as genotype information for 2,500 individuals that allowed us to characterize the mutational landscape of GPCR drug targets within an individual. And here we show that an individual harbors nearly 70 missense mutations, and probably even more given broader data sets in their GPCR drug targets. And among those, an average of 8 variants per individual have previously known clinical associations with altered drug response. Then we next seeked out to investigate what fraction of the variants that are likely to have an impact given their location. So here we've used aggregated data from the EXAC consortium, as well as all protein structures that have been available at the time, obtaining data from the GPCR DB, integrating these to identify functional sites and infer the prevalence of mutations within known functional sites. And for this, it is important to know and to appreciate that mutations can have an impact on various levels, such as on the receptor integrity, such as on the cell surface expression, stability, maybe also the trafficking and localization, and then the more sort of classical intuitive impact, like on ligand binding, on the potency, maybe on the selectivity or the kinetics, but also the signaling, the function of selectivity. Bi-signaling is very important, the constitutive activity, activation kinetics, and the internalization rates, but also on the interaction and interface to homo and heteromers or membrane interactions. And there's of course different methods and assays to probe each of them specifically. So here, given our broader functional characterization in overview, we identified that there are mutations that have an impact on different regions, and we show that 15% of all variants are actually located in known functional sites, such as the ligand binding site, effector binding site, post-translational modification sites, and microswitches. And each of those have a different impact on protein function. So we then focused on the mu opioid receptor, which is a highly variable GPCR drug target and a primary mediator of many of our opioid analgesics, and which gives rise to the current opioid epidemic. So surprisingly, we identified variants that have no evident effect on the wild-type response, which you see here in endomorphin 1, but have a significant effect upon drug exposure, such as a reduced response, such as for morphine, or even or no response for prenophin, or even a gain of function for, as in this case, for naloxone, which is not evident from its response to the natural ligand. And especially in the case of naloxone, this is it's a toxifying agent in patients with opioid overdose. That's actually quite detrimental. So if you were to identify these individuals carriers of these mutations, then you want to be informed about that. So based on that, we further developed an economic model based on drug sales from the NHS in the UK, and then estimated the possible economic impact of drug target variability, which, given different caveats, we estimated to be somewhere between 14 and 500 million British pounds annually. And these estimates are likely only the tip of the iceberg, given the expenses and the extensive use of these drugs across the globe. So, taken together, we showed that there's extensive genetic variation in drug-targeted receptors, with potential implications for drug binding, receptor activation, and signaling, and that genetic variants may lead to unintended consequences if and when treated with the drug, and that the understanding and incorporation of the effects of polymorphisms has the potential to curtail the recurrent unaccounted expenditure on public health. So the next question is then, how do we translate the genetics-informed in vitro findings into the clinics? And looking at all curated pharmacogenomics associations curated by the PharmGKB, which is a comprehensive resource that curates knowledge about the impact of genetic variation on drug response, we've seen various GPCR targeting drugs which have drug labels from FDA or from the EMA or other medical agencies that are at least informative, if not actionable or required. But actually there's no GPCR target variant labels which lead to drug labels. So if we look at the variant labels, so looking at the pharmacodynamic, so the reason why the GPCR drugs here on the left-hand side are part of drug labels is all due to the pharmacokinetics, whereas on the pharmacodynamics, there's no specific GPCR variant that is currently labeled as being actionable or required in any of the medical agencies. However, there are a number of variants where we know they have either in vitro or also in vivo implications for drug response. So the question is then, why are there so few variants in the first place that have associations to altered drug response, but also why are so few actually actionable in the clinic? And I think one explanation is that many variants are very rare and or have not been tested or exposed to medical treatment by their carriers. And this is because we have not had the access to combine genotype with phenotype data at scale as we have now. So that's what you see here, where you see here the beautiful trajectory from the discovery of the DNA double helix to the first fully sequenced genomes to a catalog of human variation to integrated health biobanks, which hold a treasure trove of information. So that now, previously, we've been really scaling up on the number of genomes we've been sequencing, but now, more importantly, we can also integrate that with healthcare records and phenotype data. And here I'm going to give, in the sort of second part of my presentation, I'm going to give a little insight into how we make use of biobank data, such as the UK Biobank, which consists of more than 500,000 participants that have been genotyped and phenotyped on various clinical measures. So in this example here, we investigated the human glucagon receptor, which is involved in maintaining blood glucose homeostasis. We performed molecular pharmacological phenotyping of 38 missense variants, so those that are changing the amino acid composition of the receptor, and specifically those we selected that are located in the receptor extracellular domain at the glucagon interface here, which you see here in red, the red peptide. And these variants were then characterized in terms of cAMP accumulation to assess G-alpha-S signaling and recruitment to beta-arrestin 1,2. And lastly, we investigated how the cAMP impairing variants and wild-type-like variants differentially impacted carriers of the UK Biobank. So on the left, you see we identify, so that's sort of a heat map of all the 38 missense variants, and on the sort of top half where it says on the right, alterations in 3, 2, and 1 pathway, these have had significant impact in at least one of the measured endpoints. So you see here the potency, the efficacy area under the curve as 3 sort of endpoints. And here you see that we identified 8 variants with decreased GS signaling, and then also performed binding experiments which revealed that the preserved glucagon affinity, which revealed preserved glucagon affinity, although with significantly reduced binding capacity. And at the bottom where it says wild-type-like, you see that we have identified a large number of mutations that had no apparent effect on GS signaling or arrestin recruitment, at least not on our cell types and assays. And just want to remember that and remind you that there is good evidence, for instance, for the melanocortin receptor, which has shown that you may not capture in, for instance, a cAMP accumulation assay all the extent a variant can impact receptor function, as I indicated also before. So we then asked the question, how do the variants that are impacting cellular phenotypes affect individuals at the clinical or physiological level? And in order to study this, we identified all carriers of these 38 tested variants in the glucagon receptor based on a quality-controlled subcohort of the UK Biobank. So here you see we started with 200,000 exome sequenced individuals, which we have quality controlled for relatedness, ancestry, genotyping quality, and consent. which we were then left with 154-ish thousand individuals, and among those we identified roughly 1,000 carriers of one or many of these genetic variants, on which we then performed whole genome regression burden testing. And doing so by grouping the CAMP affecting variants and contrasting them to the wild type like variants, we observed that the group of the CAMP affecting mutations had a higher rate of severe obesity as well as BMI and body fat percentage, while the pharmacologically wild type like mutations were not associated with increased cardiometabolic outcomes. And these observations are also in line with the role of the glucagon system in metabolism, and also support GS as the main signaling pathway affecting the physiological roles of the glucagon receptor. But also that in vitro data is indicative for real-world outcomes and can be directly linked with healthcare data. I also want to advertise here a little bit a resource that is a one-stop shop for GPCRs. Maybe some of you may have heard, called the GPCR DB, where a part of it is also a resource for genetic variants for pharmacogenomics research towards the goals of advancing this field and to fuel pharmacogenomic research with yet to be characterized variants, for instance. So please have a look here, and you also see that we have expanded recently also into G proteins and arrestin as their own database with their own tools and own merits. And with this, I think I would like to leave you with some concluding remarks. That GPCRs are still relevant drug targets, but are also a treasure trove for unappreciated human physiology and as an opportunity for personalized medicine, and that we can obtain new insights on biochemical principles and mechanistic insights from existing biomedical data that we integrate from genetics, pharmacology, all the way to healthcare data that can lead to new knowledge and potentially lead to fine-tuned clinical practices. But also that there's a huge knowledge discrepancy between GPCRs and an untapped potential across many disease areas, with many GPCRs still clinically unexploited. So what you see here is the discrepancy between, for instance, citations, but also availability of tool compounds, patents, and also protein structures. So when we talk about GPCRs as drug targets, we usually talk about a small fraction of GPCRs, but there's still many GPCRs that are both orphan but also non-orphan that can still be exploited for drug discovery. And then last but not least, I would like to thank all my supporters, funding agencies, and colleagues who have contributed to this work, especially my past and current supervisors, David Gloriam, Hans-Bräune-Osborn, Madan Babu, as well as Mette Rosenkilde from the glucagon receptor study. And I would like to thank you for your attention, and please feel free to reach out by email or Twitter in case you have any ideas for collaborations or any questions regarding my presentation. Thank you very much.